we will be starting the Unit 6. Unit 6 deals with blood test and electrical, safety and telemedicine. First is blood and instruments for clinical laboratory. The clinical laboratory is responsible for analyzing patient specimens in order to provide information to aid in the diagnosis of disease and evaluate the effectiveness of therapy. The major section of clinical laboratory are chemistry, hematology and microbiology sections and the blood bank. We have to study about certain types of blood tests used in our clinical laboratory. And before going to study about blood tests, just an introduction to the blood. For man, nature has provided a special transport system to exchange chemical products between the specialized cells of the various organs, namely the blood circulation. The circulatory system of an adult male human contains about 5 liters of blood. Blood also consists of a fluid called plasma, in which are suspended 3 different types of formed elements or blood. And these are the functions of blood, which delivers oxygen, transport waste products from cells, transport hormones, maintain body temperature, pH, fluid volume, prevent blood loss and prevent infection. And these are the basic components of blood. The basic components of blood are plasma, which is about 55% of the blood, and the remaining 45% is made up of RBCs, WBCs and platelets. These RBC, WBCs and platelets are developed from stem cells which are in the bone. This is the detailed view. 55% is made up of plasma, and white blood cells and platelets are less than 1%, and red blood cells are made of 45%. Now RBCs are otherwise called as erythrocytes. They transport oxygen in blood by chemically binding the oxygen molecule to the hemoglobin, and they are in the shape of biconcave disc without any nucleus, and their lifespan is 100 to 120 days. Next is leukocytes or WBCs. They defend body against infections and tumors. They locate areas of tissue damage by responding to chemicals. There are different types of WBCs present in the blood. They are neutrophils, eosinophils, basophils, lymphocytes and monocytes as shown in the figure. Then a cancer occurring inside the blood is called leukemia, in which the bone marrow becomes cancerous, and it consisting of huge number of WBCs as shown in the figure. Normal blood contains a limited number of WBCs. If the WBCs inside the cell becomes more than a number, it becomes leukemia. Next is platelets. They are irregularly shaped bodies. It is necessary for clotting of blood, and they are usually colorless and they have no nucleus. They are masses of protoplasm 2 to 4 millimeter in diameter. And next is blood plasma. The blood plasma with the blood cells removed is a highly viscous yellowish liquid that contains large amount of dissolved protein. It is shown in the figure, which is the blood and the plasma separated. One of the proteins, fibrinogen, participates in the process of blood clotting and forms thin fibers called fibrin. The plasma from which the fibrinogen has been removed by precipitation is called as blood serum. Next is test on blood cells. First, the whole blood is centrifuged. The blood cells sediment and form a packed column at the bottom of the test tube, which is as shown in the figure. Most of this column consists of red blood cells, with the other cells forming a thin buffy layer on top of the red cells. The volume of the packed red cells is called as the hematocrit. It is expressed as a percentage of the total blood volume. The active component in the red blood cells is the hemoglobin, the concentration of which is expressed in grams per 100 milliliter. One of the method for blood test is called as the Coulter counter method, which is used for measuring the count. It makes use of the fact that blood cells have a much lower electrical conductivity than the solution in which they are suspended. It contains a beaker with the diluted blood into which a closed glass tube with a very small orifice is placed. The placed as shown in as marked as 1. The conductance between the solution in the glass tube and the solution in the beaker is measured with the two electrodes, which is named as 2. This conductance is mainly determined by the diameter of the orifice in which the current density reaches its maximum. The glass tube is connected to a suction pump through a U-tube filled with mercury as shown as 5. The negative pressure generated by the pump causes a flow of the solution from the beaker through the orifice into the glass tube. Each time a blood cell is swept through the orifice, it temporarily blocks part of the electrical current path and causes a drop in the conductance measured between the two electrodes. The result is that a pulse at the output of the conductance meter, the amplitude of which is proportional to volume of the cell. A threshold circuit lets only those pulses pass that exceed a certain amplitude. The pulses that pass the circuit are fed to a pulse counter through the pulse gate. The gate opens when the mercury column reaches a first contact named 3 and closes when reaches the second contact named 4. Thus, counting the number of cells contained in a given volume of the solution passing through the orifice. A count is completed in less than 20 seconds. A DC voltage is produced which is proportional to the value of the count. This is one type of blood test. Next is chemical test used for blood used to test the blood. The blood serum is a complex fluid that contains numerous substances in solution. The determination of the concentration of these substances is performed by specialized chemical techniques. The commonly performed chemical blood tests are by using a spectrophotometer and a flame photo. First, we have to study about spectrophotometer. Spectrophotometry is a technique used to measure how much energy a substance absorbs at varying wavelengths of light. The basic principle of spectrophotometer is that if we examine an appropriately chosen small portion of electromagnetic spectrum, we can use the energy absorption properties of a substance to measure the concentration of a substance. Figure shows the spectrophotometer. Here, light from the light source, which is a halogen lamp, is passed through an entrance slit and incident on a concave reflector, which is usually a lens, which focuses the light on a monochromator or a prism to disperse the light. The selective wavelength from the dispersed light is obtained by taking it at the given direction and then it is allowed to incident on the reflector. From the reflector, the light beam is directed to the sample through a narrow exit slit. A sensitive photodetector D detects the transmitted light and gives an electrical output corresponding to the intensity of the transmitted light. The amplifier amplifies the output from the detector, and finally the indicator indicates the concentration of the substance. By rotating the grating, the measurements can be made at different wavelengths. This is the basic principle of spectrophotometer. That is, when a sample is illuminated, it absorbs some of the light and transmits the rest. The transmitted light IS is of lower intensity than the incident light, and it is defined as T is equal to IS by, and two beakers are placed. It is represented as I2 is equal to TI1 and I2 is equal to T square into. The light transmitted through successive cuvettes decreases in the same manner. The amount of light absorbed depends only on the number of molecules of the absorbing substance that can interact with it. The absorbance is also the same if the cuvette has a path length L but the concentration of the solution were doubled. So the absorbance can be determined by using Beer-Lambert's law. It is written as A is equal to small a into C into N. A is the absorbability, a factor that depends on the absorbing substance and the wavelength at which measurement is performed. C, concentration of the absorbing substance, and L is the path length of the cuvette. In this way, absorbability of the substance is measured. And now the instruments used in spectrometer. One is monochromator, which accepts polychromatic input light from a lamp and outputs monochromatic light. It consists of an entrance slit, a lens or a mirror, a dispersion element, focusing lens, and exit slit that disperses the white light into different colors and different. And then cuvette means it is a design to hold samples for spectroscopic experiments. It can be made of plastic, glass or quartz. It should be as clear without impurities. Then detectors, which convert radiant energy into electrical signal. The photocell and phototube are simplest photo detectors. Then display device, the data from a detector are displayed by a readout device such as an analog meter, a light beam reflected on a scale, or a digital display or LCD. The output can be transmitted to a computer or a printer. These are the different types of spectrophotometers used today. Thank you.